Hello viewers, today we will talk about major histocompatibility complex. What is this complex? It's just a group of genes, and this group of genes code for certain proteins. And these proteins are found on the surface of cells. And proteins are found with this major histocompatibility complex is coding for these kind of proteins. So these major histocompatibility complexes are present in almost all vertebrates. And what is their role? Their role is to help immune system in immunosurveillance. So these are the ones which are helping to recognize foreign from that means self from non-self too. In case of human beings, it is called human leukocyte antigen system 2. Either we call that it a major histocompatibility complex or human leukocyte antigen system. So next time when you hear about HLA, it's the same as MHC. This MHC is clustered around chromosome 6 in case of human. This chromosome 6 is the place where you can find these group of genes. And this group of genes code for different proteins, and these proteins are usually not common between two human beings. Rarely, it's reported that they are not very common, and that is why histocompatibility word is used. That means tissue types. So two humans will have two different kind of major histocompatibility complexes, and so their tissue types will be different. Some of these genes also code for, that means some of the genes of major histocompatibility complex also code for complement proteins, cytokines, and certain kind of enzymes. So they are called class 3 MHC molecules. Class 3 MHC molecules may not be so well known and common, while class 1 and class 2 are the ones which are studied more. Class 1 are assembled in endoplasmic reticulum, and it is present in normal healthy cells. And what class 1 molecules are doing, they are the beta and alpha and beta chains, and they have a groove. In that groove, they take all the peptides which are formed in a cell, and they take that peptide in that groove and take it out of the cell, show it as a flag to T cells. And when that flag is shown, T cells knows it is self. There is an understanding that this is in a normal cell and this is a normal peptide of a normal cell. So T cells are not activated, and T receptor cells do not bind with it, and hence no process takes place. So in case of MSC1, they show all the normal proteins, peptides present in a normal cell, and they just flag it out, while the roaming T cells do not get activated, and T cell receptors do not fix in that groove of MSC class 1 when normal things are going around. And this MSC class 1 gets activated in the presence of tepacin. This enzyme name I'm telling for the people who are going for the entrance exams, the tepacin is the one when MSC class 1 gets activated. And in case of infected cell, what happens that we are going to read, it's in this mentioned in the slide, but this will be very clear when we will see it in a diagram. So I will talk about it in a diagram. First, let's talk about MSC class 2. MSC class 2 is normally found on antigen presenting cells, whereas MSC class 1 is present in every healthy cell. MSC class 2 will be seen in antigen presenting cells. What are antigen presenting cells? I have given a full lecture on that. If you want, please go and see that video too. Then this antigen presenting cells present processed antigen. That means when antigen presenting cells engulf and then they process the antigen, and there comes MSC class 2. This antigen is fixed in the groove of MSC class 2, and then they take it to T helper cells. How all this happen? This will be clear in the diagram when I will be showing it in a diagram. So this MSC class 2 has a very important role in initiating adaptive immune response. Remember, MSC class 2 has an important role in initiating adaptive immune response, that is proliferation of T cells and B cells, and then synthesis of immunoglobulins, secretion of immunoglobulins. While MSC class 1 is the one which is showing normal proteins, and if a cell becomes abnormal, that can be because of cancer or tumor, then it takes that to cytotoxic cells. So MSC class 1 takes the things to T cytotoxic cells, while MSC class 2 takes the things to T helper cells. This will be very clear by this diagram. This is if a normal cell gets infected, what happens? This antigen fragment, that is because of the infection, that fragment will be put into this. This is the MSC class 1. This will be put into the groove of this MSC class 1. And once it will take that infected fragment here, this infected fragment, T cell receptor of cytotoxic T cells will immediately recognize it as foreign, and they will come and start the process of apoptosis. So this happens in the case of MSC class 1. What happens in the case of MSC class 2? MHC class 2 is the one which are present in antigen presenting cells. They are not present in all the normal cells. So when antigen presenting cells are usually macrophages, dendritic cells, B cells. So this microbe, when enters into the antigen presenting cells breaks this microbes into different kind of and into the different fragments. That means they will process the antigen. That is why they are called antigen presenting cells. They will process the antigen, and this processed antigen will fix into the MSC class 2, groove of MSC class 2. And this MSC class 2 will then take it, flag it out and show it. And there the T helper cells will come, get activated, and these helper cells receptor will receive this MSC class 2. So this is the functional difference between MSC class 1, MSC class 2. MSC class 3 are the ones which helps in secretion of certain complement activating system and all. More work is not available in MSC class 3. What we have to concentrate is MSC class 1 and MSC class 2. MSC molecules have important role in transplantation. Till now, you must have understood when MSC class 1 is activating and when MSC class 2 is activating. When allografting is happening, that is transplantation is there, any tissue organ. So MSC molecules know self, and they can differentiate self from non-self. MSC class 1 is the one which is present in normal cells, and it is always exposing the endogenous molecules, endogenous peptide fragments. Is always exposing endogenous, that is present in the cell, in a normal cell. But when tissue is transplanted or organ is transplanted, it has its own MSC. And this MSC will be different from the one, it will have its own molecules. This will be different from the one present in the host. So when host MSC will come across that peptides, it will take it out and flag it out. And once it will flag it out, then T cytotoxic cells may come and start the process of apoptosis. And so rejection of transplanted organ or tissue can take place. So MSC, study of MSC is very important. It play an important role in organ transplantation and accepting that organ or tissue. So it has an important role, and certain autoimmune diseases also happens because of the problem in this MSC. So that we will be reading in future classes. Thank you so much.